about mitosis and meiosis. We were talking about the difference between mitosis and meiosis. Meiosis is two cell divisions, right? Mitosis goes 2N, you wind up with 2N. Meiosis, you go 2N, you wind up with 4 1N cells, right? The first cell division is a reduction division. So you reduce the number of chromosomes, and it depends how you define the term chromosome, right? So you should make sure you have that definition correct. When we talk about this thing about in meiosis, there's a special event which hopefully you are aware of by now. It's called crossing over, and some people call it recombination. And we'll go over this in a little more detail conceptually here today. We'll talk about the effects or practical reasons why it's important over the next couple of lectures and how you can map all of these genes out and such using recombination. When we get into the next section of the course, we talk about DNA replication. We talk about how DNA polymerase actually copies the DNA. And we also talk about crossing over in molecular detail in that section. Okay? So, this is such an important event that in prophase 1, this is where it happens. In a normal situation, the only time crossing over will occur is in prophase 1. There have been demonstrated that on very, very rare instances, in an apparent screw-up, a cell which should not be engaging in crossing over does, or has performed that by accident. In general, this is preserved as an event that only happens in prophase 1, that first cell division of meiosis. It does not happen in mitosis in a normal, healthy situation. The event is so important that prophase 1, the same way we draw this out, we've got G1, S, G2, and M, and we break M down into prophase, metaphase, anaphase, telophase, cytokinesis, right? We can now break this down into smaller subdivisions. Prophase 1, right? So, leptotina, zygotina, pachytina, diplotina, and diakinesis. There are very specific events that happen at each one of these stages. But basically, the chromosomes start coming together. In leptotina and zygotina, they form the synaptonemal complex. And it's illustrated here as the two chromosomes sort of come together and touch. We'll see another image of this. The precise mechanism, I know functionally what happens. I can't tell you what molecules come in and make it happen. But basically, if you have dad's chromosome and mom's chromosome, so they're the homologous chromosomes, there are proteins here, and I should do it a different color. There are proteins here that bind to some specific sequence. They bind here. These proteins will match up so that this gets dragged next to this. And the proteins physically interacting with each other hold these guys together, like this. Does that make sense? That is the synaptonemal complex there. In pachytina, actually, let's go through, show each one of these steps individually here. The crossing over occurs in pachytina. Diplotina, it kind of resolves, and we're left with this structure. Originally, I thought this is when the DNA exchange occurs, but it actually occurs here. Where's this here? Crossing over. Okay, so here's our picture. Whoops. I'm going on mine, not my bad. This is the synaptonemal complex. This represents one strand of DNA. This is the other strand of DNA. These are the proteins. So the same way that you can blast search the NCBI and say, where's the sequence, right? Basically, homologous sequences line up so that they're basically, literally, base pair per base pair matched perfectly for our purposes right now. get into a later section of the course, we'll talk about how you get some variation going on there. This is an electron microscopic image where they show the DNA here, and you see the protein bands here that are holding them together. In Pacatina, the sugar phosphate backbone actually breaks. So let's draw this out the way I do. I've got my sugar connected to a phosphate, connected to a sugar, to a phosphate, to a sugar, to a phosphate group, right? This is the backbone of DNA. The bases poke off here. And they form their hydrogen bonds with the other strand that's going anti-parallel this way, right? The sugar phosphate backbone actually breaks like that. That covalent bond is gone. Okay. And so on. What happens molecularly is this covalent bond comes over here and breaks this guy. So that this guy can come over and connect like that. This is why it needs to be lined up precisely nucleotide to nucleotide. Crossing over is a physical trading of DNA. Make sure that's clear to everybody. So as this crossing over is going to occur at the Synaptonemal Complex, when the crossing over is completed, we will have a chromosome that is this. Gesundheit. So that where the Synaptonemal Complex lined up, DNA has physically been taken off of here and put on here, and DNA has physically been taken off here. They physically trade the DNA. If the purple was a radioactive piece of DNA, and this was not radioactive, this now has a small portion of it that is radioactive. The atoms that were here have been physically transferred onto here, okay? Yeah.
Is there always a specific, like in humans, is there always a specific part that's crossed over?
Yeah, that's great. So I was thinking about that as I said it. So proteins bind to these sequences and draw them together. And it makes, that would lead you to then assume that there's only certain spots in the human genome where this occurs. And that's not the case. It happens randomly, apparently randomly, anywhere in the human chromosomes.
That's pretty.
There appear to be hot spots where it happens a little more frequently than others, but it can happen anywhere. So there's no consensus sequence that says, this is where it occurs. This is where the proteins bind to make this happen. Thanks for that question. So this is called the chiasmata. And basically, once the exchange has occurred, the chromosomes are just going to hang out. They're just kind of hanging on each other. The proteins that have performed the operation of splicing the DNA, changing that's making these Frankenstein chromosomes, if you will, those proteins kind of hold them together for a little bit. And eventually they'll separate. And that's diakinesis, and the physical exchanges occur. Any questions on crossing over?
So what is the chiasmata? Is that just like...
The chiasmata is when they're kind of laying on top of each other like that.
Okay.
Yeah.
So the whole point of the protein is just to line them up so that crossing over can occur at some other point along the chromosome? Where it's
The Synaptonemal Complex is where the crossing over is going to occur. Okay. And actually, when crossing over is going to occur, the proteins actually, the whole chromosomes pretty much line up.
Yeah.
All of the proteins pretty much line up. But the Synaptonemal Complex kind of defines you can cross over at
Because like
these
other
nucleotides.
pictures, it looks like the chiasmata is a different place in the Synaptonemal Complex.
Oh, I think it starts here. I guess what they're showing is it starts here and it severs its way up. So I guess my impression of it was different. But I've always, well, when I learned it, this is where the crossing over would occur. I guess
Okay.
they're showing it different. I guess I don't know the details on that. Maybe they know more about it now than I am. Sorry about that.
I'm sorry.
But yeah, here they show the whole chromosome kind of zippers itself together and lines itself up. We'll talk about errors in that process later on. Okay, other questions about this? So, why would this occur? Evolutionary advantage. So if we think, we typically think of blonde hair, blue eyes going together. So those alleles going together. So if the allele for blonde hair is here, and the allele for blue eyes is here, by having this crossing over, you can now mix things up such that now the blue-eyed allele would be with brown hair, and the brown-eyed allele would be with blonde hair, right? So practically, that's what happens. By changing the DNA, you get different sequences mixing and matching together. You get different combinations, right? Because we all know that brown hair and blue eyes is the best combination. Okay. Okay. So we've already gone over the reduction division discussion. And so I think we can finish this out. So in males, meiosis produces 4 identical cells, which basically then develop into the sperm, 4 gametes. In human females, what happens is the divisions are unequal. So in meiosis 1, one cell gets almost all of the cytoplasm and all of the resources. And the other cell that pinches off is this tiny, dinky little cell, and it's actually, it survives. It doesn't really perform much of a function. That's under some debate. It's a very small cell. We call it a polar body. This cell then is going to undergo a mitotic cell division. They don't show it here. But it actually divides into 2, and you get these 2 tiny, tiny little polar bodies. And as this guy goes through meiosis 2, it divides, and it keeps, again, one cell keeps almost all of the cytoplasm and all the resources, and a smaller little cell pinches off and becomes yet another polar body. So at the end of meiosis in a female, there's one very large cell, that's one end, we call it the egg. There's a small polar body, and then there's 2 really small polar bodies. And what I learned is those polar bodies do nothing, and they fade away and are gone. There's some debate, there's some suggestion that they hang out with the egg, and they feed hormone signals to the egg to tell it, hey, you're an egg, you're ready to get fertilized, and to kind of support it.
These occur like in females when you're just like a kid, and that it doesn't self-happen.
Yeah, so this process happens in females, and so Kate is 5 and Ellie's coming on 4, so they have all of the eggs that they're ever going to have. Until about 2 years ago, that's what we would have said. It now looks like adult females at very low levels can have cells start to go through this process and can generate new eggs. But in general, each ovary has about 1 million of these already at birth. They're already made and ready to go. Okay, I put this up here. These are the key points, the key differences between meiosis and mitosis. So I've tried to summarize this on here, right? I'm not going to run through this. This is for your own study. Okay. Might be important, this slide right here, for maybe a long essay, if we should have an exam on this, right? Okay, just for fun, let's calculate this out. So my brother is older than I am, and we can say, you know, my brother so at one point, it's kind of funny to say, I hope you guys are adult enough college kids that we can talk about this. At one point, my brother was an egg and a sperm that came together, right? So for that egg to form, say there were 23, this is my mother's 2N cell, and as meiosis went, the chromosomes segregated to make a 1N egg. So this egg got some pattern of these. In this case, it might have taken this chromosome here, and then it took this chromosome, let's say it took this chromosome here, and then it took this chromosome here, right? And we'll say the purple one again here, and then we'll do the black one here, right? So in my mom got this from my grandfather, and got this from my grandmother, right? So my brother, the egg that came to be my brother, had some mix of those, right, from both my grandparents, right? And then in the sperm that was used to create my brother, there was some combination from my parent grandparents on the other side, right? Right, like this. What are the odds that the egg that formed me got all the exact same chromosomes from my mom that the egg was for my brother? Does that make sense? You know how you calculate that out? That's up here. So there's a 1 in 2 chance. So we know what Scott's genotype is. We know what chromosomes he got. There's a 50% chance that I got this same chromosome. And there's a 50% chance that I got this same chromosome from my mom, right? There's 23 chromosomes. So it's 2 to the 23rd, which is 1 in 8.4 million. And then, what are the odds that the sperm that created me would have gotten the exact same combination of chromosomes from my dad? Same thing, right? Not exactly, because I'm a guy, my brother's a guy, so we both got the same Y. And that had to happen, right? So it's actually 2 to the 22nd, right? Because we know we both got the same Y. But I think I have 23rd up here anyway. So for females, this would be the correct thing. So the odds that 2 eggs would have been created in my mom that had the same assortment of chromosomes, and 2 sperms would have been created that had the same assortment. And then the odds that this one would find this one, so that I could be an identical twin of my brother, born from a different pregnancy, right? So that's 8.4 million times 8.4 million. Does that make sense? So the odds of identical twins, genetic identical twins being born, we'll get to that in a second, being born in different pregnancies, so years apart, just by this random chances, what is that number? Billion, 70 trillion? 1 in 70 trillion.
And does this not account for like crossing over?
Right. So, as improbable as this is, crossing over actually occurs such that this would kind of mess this up, right? Because in meiosis, crossing over occurs. Like that. And the crossing over that would happen in the process of forming the egg that formed my brother is not going to crossing over isn't going to happen the same way in me. So there's really no chance of identical twins being born that way. I don't think there's been 70 trillion humans on Earth over the course of what we would call human existence.
So not yet.
Not yet. Yeah.
There's still a chance.
Get busy, guys.
Don't think we can do that.
So in humans, on average, this is an average, crossing over happens on every single chromosome one time. So every egg formed, on average, each chromosome has crossed over one time. Sometimes the chromosome will have crossed over 2 times or 3 times. Sometimes the chromosome won't cross over at all. But on average, one. There's a lot of genetic mixing that happens. Pretty cool, isn't it? Yeah.
Does crossing over like take the whole genes, or is it like you were saying it's kind of random, right? So are there like the whole gene for blue eyes? Like, is that completely crossed over?
Yeah. Do genes go in segments?
Yeah.
That's a great question, and I think let me break your question. Let me ask your question a different way. Do the machines that perform the crossing over know the boundaries of genes,
Right.
so that they protect and they only go in between genes? And the answer is no. They're machines. They don't have a brain. Wherever they bind to the DNA and do it, that's where they're doing it. That said, the human genome, if you have a chromosome, if you guys go back to the NCBI and you look at all the genes in the chromosome, you might have a gene here, and you might have another gene down here that they say, and this might be 1 million base pairs there. And if this is 50,000 base pairs, sorry, just 50,000, I got it right bigger for these people over here. The odds of crossing over happening in the middle of a gene is much less than the odds of crossing over happening between genes. Right? So it happens right in the middle of genes.
You could like have an intron that would get sort of messed up too, that would have an effect, but what's the same types of intron differences?
Ask that again.
So
I got in real late. I'm really, really tired.
we have 2 exons. There's an intron missing.
Okay, you're stealing the thunder from me about 2 weeks from now, but yeah, let's do this. This is great.
So if there was crossing over in some region in the intron that was like somehow important, maybe right at the end where a consensus sequence might be, could that have adverse effects?
Right? Okay. So what you're saying, so let me get this going here. Okay. So, say that again. You have crossing over where?
If, say, in between those two little, or if any of them,
Okay, so do crossing over right here.
could that have an effect on like the expression of the genes? Or are there
Yes.
sensitive sequences in there that could get screwed up?
Sure, yeah. So in here, so what we've got is we've got our CAG, TRAGT here. And we've got our CAGG here, right?
Yeah.
CAGG, I missed that one. Right? So that's how it cuts. Somewhere in here is a branch point A, is the branch point adenine, right? It's usually going to be like right about here. And so there's a consensus sequence in there. There are probably other sequences in here that help drive splicing that we haven't found yet. Yeah, if you get crossing over here, you might switch those things around.
And then you would have a non-functional gene, or
Well, I mean, in this case,
it still has some.
we'd have all of
function.
this code, so we'd be losing the tail end of it, possibly. But remember, when this goes away, you're going to replace it with, and hopefully, the code for this isn't too far off of whatever allele this is, isn't too far off of what this is. Right? But if this thing has unique, interesting mutations right here and here that do really good things, and this guy has a unique, interesting mutation here that does good things, by recombining in the middle of the coding part of a gene, we can get this really good mutation lined up with these, and you get like super allele. Glowing blue eyes, right? So, stealing my thunder for later in the semester. Do you understand why it would be valuable now to have coding parts of the gene in little things with huge spaces of intron in between? if this intron is 50,000 base pairs, you're burning 50,000 ATP every time you transcribe it. You can cut that out and throw it away. But potentially, the value of putting together different mutations in a gene over evolutionary time, it's worth spending all that ATP to get different mutations together to get the glowing blue-eye gene. Right? Does that make sense? So the reason we have small little exons and huge introns, this is a concept called exon shuffling. It allows you to move things around very easily. And we'll take the exon shuffling concept to a whole nother level when we get later in the semester. Awesome. Well, Alex was excited by it. You guys are like totally bored.
I think this is really cool.
Yes. Alex is right. You get the gold star today. Sweet. Okay.
You can get it taken away.
By
By the way, people.
the way, people, you can get your gold star taken away. Very good. How do you know that?
It's an emotional process.
Okay. So let's talk about this genetic linkage. It was first really identified in studies on Drosophila. So here's the example we'll run through. So first of all, let's talk about the sex chromosomes, the X and the Y. So in humans, we have the XY system. An organism that is XX, female. Organism that is XY, male, almost always. That's the case. You can get, so the X chromosome and the Y chromosome, they do not have homologous sequences, mostly. There are a few spots on the Y that do match the sequence of the X, and sometimes you can get a synaptonemal complex to form between them, and you can get crossing over such that the genes for male get stuck on an X, or the genes for male get taken off of the Y. And you can wind up with someone who has an X and a Y, but doesn't have the male imparting genes in the Y. And so they can't develop as male. Okay? Which is dominant? Is male dominant over female?
Yes.
The heterozygote is XY is male, right? We can also tell, so it could be actually two ways. You could say two X's direct formation of a female, or a Y directs formation of a male. There are people who exist on Earth that are XXY. Through a non-disjunction event, chromosomes don't segregate properly. The person winds up inheriting two X's and a Y. Usually the two X's come from the mother. It's possible that an X would come from the father, but usually it's from the mother. And an individual who is XXY develops as male. Not all organisms follow the human XY process. Birds are the opposite. And they go on the ZW system. So an organism that is ZW is female, and the organism that is homozygous is male. Does that make sense? Who's taking the MCAT here shortly? A couple of you? I will give you a 90% chance that they will ask you about they will ask about sex-linked genes and they'll use birds as their example because it's backwards, right? I want to see if you can think through that. If you're going to take the MCAT, I recommend you study and prepare for this. I bet there's probably a paragraph and 5 questions on it. Okay? So, it operates basically the same way, follows Mendelian genetics. You inherit this stuff, you know, the chromosomes segregate the same, it's just that the homozygous is male and the heterozygous is female. Some organisms use an XO system. Something that is XX is female. Something that is X and doesn't have another something is male. In other cases, here's one. an egg that gets fertilized and is 2N develops as a female. An egg that never gets fertilized develops as a male and is haploid. And that is bees are an example of that. So there are some variations to the sex chromosomes. So how did we learn about this sex chromosome linkage stuff? So there was this very famous researcher around the turn of the century, was kind of repeating the Mendelian genetic stuff and said, all right, I'm going to take this fruit fly that I just found flying around my bananas and this one over here, and I'm going to take this and I'm going to breed it, inbreed it for a number of years until I am absolutely sure that I have a pure breeding line of whatever this fruit fly is. And then I can start to cross with other flies and do my experiments this way. So this person, a generation time for a fruit fly is about a month. So this person had maintained this pure breeding culture for many years, 2 years, 3 years, and all of the flies were identical. It was a pure breeding line. One day, he had anesthetized the flies and was picking through them, and there was a fly in his population that had white eyes. And all of his other flies were pure breeding, wild-type red eyes. So somehow, in his tube, a new mutation must have come to exist in the population that screwed up the eye color. Or he contaminated, but we'll assume that he was working very carefully, right? So, it doesn't show very well here, and I'm not going to turn the light on, but red eyes, they look like, they're not quite this color red. We describe them as brick red, not quite this color red. They're about the color of Kevin's shirt there, maybe a little bit, with a little bit of hint of a darker like that color in it. And white eyes are absolutely devoid. They are a real bright white. They're devoid of any color. So he decided, I'll study this new allele, this new version of this gene that I just found here. He took white-eyed males and crossed them with a wild type, so a red-eyed female, and all of the progeny, all of the F1 had red eyes, which is dominant. Red eyes is dominant over white eyes. Okay? He then self-crossed these, and all of the F2 females had red eyes, and half of the F2 males had white eyes. Whoa. What are the odds that you would have a Punnett cross or a Punnett square lined up like that? Like this. It's pretty simple, right? And all of the females happen to be one of these, and none of these. And half of the males were this, and the other half were these. Not likely to happen. Just by chance. He was breeding up, I mean, you get to thousands of flies when you get to the F2. So it looked like the eye color gene was following and going along with the sex genes, sex determining genes. So let's draw this Punnett square out here. Let's show what happened. I need to see the crossing. White-eyed male. And a red-eyed female. And this is the way I do it. Okay? People use different numbers. Some people will say, that's red, and that's white. I think that's what I have in the PowerPoint file. I do it this way. Okay? Why am I putting it on the X chromosome and not the Y chromosome?
It's not all the males in here.
Not all the males inherit it? The Y chromosome is really, really small and there's almost no genes on it. So if something is sex-linked, it's going to be on the X. The X is a full-size chromosome. Lots of genes on it. Y chromosome just doesn't have many genes. That's why. So we'll make that assumption for all sex-linked crosses, okay? Okay, so what happens is we can wind up with an organism must inherit this, right? And it can either inherit this, or it could inherit this. So this guy also has to have inherited this, right? So this is the P, the parental. This is my F1. All of the flies have red eyes. Perfect, awesome. Now let's cross this. The only cross we can do is this with this, right? Because you can't have this self-cross with itself, right? And this can't self-cross with itself. Turns out we get 3-to-1 ratio. 1 white, 3 reds. We're still following Mendel's laws. We're still segregating this way, right? Still get a 3-to-1 ratio. All of the females have red eyes because this is a big R. These are pure breeding and these are carriers, heterozygotes, and half of the males have red eyes and half have white eyes. Right? I want you to do the reciprocal cross here in class right now. So I'll set it up for you here. White-eyed female. Oops. With a red-eyed male. What is the F1? What is the F2? You guys ready for me to start on the F1? Who wants me to not do the F1 yet? Those are the two possibilities. All of the males have white eyes. And all of the females have red eyes. Okay. Let's do my Punnett square. I'll give you guys a couple more minutes on this. Ready? Am I doing this right? You gotta check me if I'm doing this right. Yeah. half the females are white-eyed, half the males are white-eyed. Is that right? And the others, so there's no, I guess the white eyes are pure breeding here. They have to be because they're recessive, right? All right, I want you to be able to do this on the exam. I want you to be able to follow a sex-linked trait like this on the exam, okay? So I have a bunch of slides here that show this. I can run through that. That's just this right here. Okay. Before we get into genetic linkage, I think this is a really good stopping point. So before we get into this, we're just going to take a break here. We'll come back to this. We'll start this next time. We'll talk about sex-linked process. We'll talk about genetic linkage in general next time. We'll run through a few examples. All right. Thank you very much. We will definitely meet the lab today. If the primers are not yet here, we'll just go to the classroom and we'll talk through the process. Hopefully the primers are here. We can walk through the process and we can actually start doing some wet lab stuff today, okay?